Hey guys, in today's video we will be starting with the topic endomembrane system of the chapter 8 Cell the Unit Life. Membranous organelles are different in terms of their structure and function. Many of these are considered together as an endomembrane system as their functions are coordinated. The endomembrane system consists of endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. The functions of mitochondria, chloroplast, and peroxisomes are not coordinated with the endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. These are not considered as part of the endomembrane system. Now let us study the different components of endomembrane system in detail. Starting with the endoplasmic reticulum. The endoplasmic reticulum is a network of tiny tubular structures scattered in the cytoplasm. Endoplasmic reticulum divides the intracellular space into two different compartments, that is luminal, which is the inside endoplasmic reticulum, and extraluminal, that is the cytoplasm. Ribosomes are attached to the outer surface of the endoplasmic reticulum. Endoplasmic reticulum, which bears the ribosome on its surface, is called as the rough endoplasmic reticulum. The absence of the ribosomes on the outer surface of the endoplasmic reticulum is called as the smooth endoplasmic reticulum. Endoplasmic reticulum are extensive and continuous with the outer membrane of the nucleus. They help in protein synthesis and secretion. Smooth endoplasmic reticulum, animal cells lipid-like steroidal hormones are synthesized. Whereas smooth endoplasmic reticulum helps in the synthesis of the lipid. For instance, in animal cells, steroidal hormones are synthesized due to smooth endoplasmic reticulum. Talking about the ribosomes, these are the granular structure which was first observed by George Palade under the electron microscope as dense particles. Ribosomes consist of ribonucleic acid RNA and proteins and are not surrounded by any membrane. The functions of ribosomes are they help in synthesis of proteins. Proteins are needed for many cellular functions such as repairing the damaged cells or tissues or directing the chemical processes. Ribosomes can be found floating within the cytoplasm or attached to the endoplasmic reticulum. Now let us study about Golgi apparatus in detail. Golgi apparatus was first observed by Camillo Golgi in 1898. They appear to be stained reticular structures near the nucleus. They were later named as Golgi bodies. These bodies consist of many flat disc-shaped sacs or cisternae. These were arranged or stacked parallel to each other. Different number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with different convexes or the forming phase and concave trans or the maturing phase. Some of the functions of Golgi bodies are proteins synthesized by ribosomes on the rough endoplasmic reticulum are received by the cis phase or the forming phase, which later on is modified in the cisternae of Golgi and are packed in the form of vesicles and released from trans phase or the maturing phase. Packaged materials are delivered to intracellular targets as well as secreted outside the cell. Golgi complex produces lysosomes. Golgi complex is also the site of formation of glycoproteins and glycolipids. Coming to the next topic, that is lysosomes. Lysosomes are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosome vesicles have been found to be very rich in all types of hydrolytic enzymes such as lipase, proteases, carbohydrases, etc. Lysosomes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Next is centrosome. Centrosome is an organelle containing two cylindrical structures called centrioles. These are surrounded by amorphous pericentriolar materials. The centrioles lie perpendicular to each other, in which each has an organization like the cartwheel. These centrioles are made up of 9 evenly shaped peripheral fibrils of tubulin. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the centriole is also proteinaceous and called the hub. It is connected with the tubules of the peripheral triplets by radial spokes made of protein. Centrioles forms the basal body of cilia or flagella, and the spindle fibers gives rise to the spindle apparatus during cell division in animal cells. Next are vacuoles. Vacuoles are the membrane-bound space found in cytoplasm. It contains water, excretory products and other materials which are not useful for the cell. Vacuoles are bound by the single membrane called tonoplast. The tonoplast in plants helps in the transport of number of ions and other materials against the concentration gradients into the vacuole. This is the reason why the concentration is significantly higher in the vacuole compared to the cytoplasm. The contractile vacuole present in amoeba helps in excretion. In many cells, such as in protists, the food vacuoles performs the capturing or engulfing the food particles. That is all about the endomembrane system and its components. Now the next topic is mitochondria. Mitochondria are a double membrane structure. Its inner membrane is folded many times and forms cristae. The space between the two membranes is called as perimitochondrial space. The inner surface of the inner membrane of the mitochondria, there are knob-like particles called as Racker's particles. They help in transport system. The mitochondrial matrix is the granular fluid-like substance consisting of the prokaryotic ribosomes, DNA, and also several enzymes which are necessary for the Krebs cycle. Functions of mitochondria are: it is the site of aerobic respiration and ATP generation. That is the reason why they are called as the powerhouse of the cell. Mitochondria also provides different intermediates for the synthesis of biomolecules like cytochrome, xanthocyanin, etc. The last topic of today's video is plastid. Plastids are the pigments which contain cell organelles that has double membrane envelope. These are found in the plant cells. There are 3 types of plastids, namely chloroplast, which are green in color, chromoplast, which are yellow, orange, and even red in color. Leucoplast are colorless plastids. Plastids are not only found in plant cells but also in euglenoids. Chloroplasts are found in plant cells, especially in the leaf, as they contain chlorophyll and carotenoid pigments which are responsible for trapping the light energy that is required for the photosynthesis process. Chromoplasts consist of carotenoid pigments like keratin, xanthophylls, and also other pigments. Other plastids includes amyloplast. They store carbohydrates such as starch, for example potato. Then alloplasts, they store oils and fats. Aleuroplasts are the another type of leucoplast which stores proteins. So the leucoplast, which are colorless plastids, consist of amyloplasts, alloplasts, and aleuroplasts. Major number of the chloroplast of the green plants are found in mesophyll cells of the leaves. Chloroplast is bounded by two membrane. The space between the two membrane is called as the periplastidal space. The space limited by the inner membrane of the chloroplast is called as the stroma. It contains colorless watery fluid and also enzymes of photosynthesis, circular DNA, RNA, and also ribosomes. Stroma is the site of dark reaction. Stroma consists of membrane-bound flattened sacs called thylakoids. They are arranged one above the other, which forms granum. Thylakoids contain photosynthetic pigments like chlorophyll, carotenoids, xanthophylls, etc. Grana are the site of light reaction. So stroma is the site of dark reaction and grana are the site of light reaction. Function of the chlorophyll is that it is responsible for the photosynthesis process. That's all in today's video. Thank you.